Welcome to the Biomedical Podcast, the podcast that connects groundbreaking research with real-world answers. I'm Dr. Bensada, and in every episode, we examine new published articles to take a close look at the discoveries that are changing how we see science. We aim to fire up your interest and start a talk that goes beyond the lab.
Welcome back to the Biomedical Podcast. And apologies first, we know we missed you last week, but we're back today.
Yes, good to be back.
And we're diving into something really fascinating. Small interfering RNA, or siRNA. You've probably heard the term buzzing around. Today we want to unpack what this actually means for treating all sorts of things, right? From viruses to cancer, even genetic conditions.
Exactly. And we've got some great material to draw on clinical trial results, scientific reviews, studies on how these are actually being used.
So the plan for this Biomedical Podcast is to give you, our listener, a really clear picture of siRNA without getting bogged down in jargon. What's really important here?
Let's start with the fundamentals.
Okay, so what is siRNA, small interfering RNA?
Right. So at its heart, it's a tiny molecule, a really tiny molecule, but with a huge job. It can silence specific genes.
Silence them how?
It works after the gene has been transcribed into messenger RNA or mRNA. Think of it like intercepting the instructions before they can be carried out.
Yeah.
A very precise off switch.
A specific gene off switch. Okay, that sounds powerful. How does it work in the cell? Is it a natural thing?
It actually taps into a natural process called RNA interference, or RNAi. You might remember the Nobel Prize for this went to Andrew Fire and Craig Mello back in 2006.
Yes. For discovering this whole mechanism.
Exactly. They found that double-stranded RNA could trigger this gene silencing. And siRNA is a short piece of double-stranded RNA, usually around, 21 to 23 nucleotides long. Very small.
Okay, tiny double-stranded RNA enters the cell. Then what?
Well, first, it usually gets processed by an enzyme called dicer, like molecular scissors, chopping it to the right size if needed.
Dicer, got it.
Then this little siRNA molecule gets loaded into a protein machine, basically. It's called the RNA-induced silencing complex, RISC for short.
RISC. Okay, so the siRNA is now part of RISC. What's its role there?
Inside RISC, the two RNA strands separate. One strand, the guide strand, sometimes called the antisense strand, stays with RISC.
The guide strand, and it guides RISC.
Precisely. Its sequence matches the messenger RNA of the gene we want to shut down. It's like a homing beacon. RISC uses this guide strand to find the target mRNA floating in the cell.
And when it finds it?
It destroys it, chops it up. If there's no mRNA instruction sheet, the cell can't build the protein that the gene codes for. So the gene is effectively silenced.
Wow. That is incredibly specific.
It is. That specificity is really the key advantage compared to, older drugs that might hit multiple targets.
But it sounds almost too neat. I mean, getting these siRNA molecules to the right place in the body working properly, that must be tricky.
Or absolutely. There are some major hurdles. First, siRNA molecules have a negative charge. Cell membranes are also negatively charged.
Like charges repel, so getting inside the cell is hard.
Exactly. Plus, our blood is full of enzymes, nucleases that just love to chew up RNA. So the siRNA needs protection.
Okay, charge problem, degradation problem, anything else.
And just getting it to the right cells. You don't want liver siRNA ending up in your brain, for example. You need targeted delivery.
Right. It's delivery systems. That sounds critical. How are researchers solving this?
Yeah, this has been a huge area of focus, developing clever ways to package and deliver the siRNA safely and effectively.
What kinds of packages are we talking about?
Lots of different approaches. Nanocarriers are a big one. Tiny, tiny particles that wrap around the siRNA.
Nanocarriers? Like what?
Lipid nanoparticles, LNPs, are probably the most well-known. They've been crucial for some of the approved siRNA drugs already. You might have heard about them with the mRNA vaccines too. Similar tech.
Okay. LNPs, are there other delivery methods being tried?
Oh, yes. People are using aptamers. Those are short DNA or RNA bits that bind to specific cell surface markers, like a key finding the right lock.
Lever.
Also peptides, short protein fragments to help cross cell membranes, even things like special sugars, other proteins, antibodies. Basically trying to give the siRNA a delivery address and a protective shield.
It's amazing molecular engineering. Okay, let's connect this to diseases. COVID-19 is still fresh in our minds. Was siRNA explored there?
Definitely. When the pandemic hit, there was an urgent hunt for treatments, and siRNA looked promising.
Why specifically?
because you could design it to target parts of the SARS-CoV-2 virus genome that don't change much between variants, the conserved regions.
Ah, so potentially a treatment that could work even as the virus evolves.
That was the hope, yes. A broad-spectrum antiviral approach.
Any specific examples that went into trials?
There was one called SNS-A12. It was an inhaled siRNA, chemically modified for stability, and it targeted a really crucial viral gene called RDRP.
RDRP, RNA-dependent RNA polymerase, the enzyme the virus uses to copy itself.
Exactly that one. Stop the copier, stop the virus. So they did a phase 1 trial in healthy people, delivering it as a nasal spray. Chang published on this in 2025.
And how did that early trial go, safety-wise?
The results looked pretty good. It seems safe and well tolerated, both as single doses and multiple daily doses for a week.
Did it get into this? Okay.
Yeah, they found it was absorbed pretty quickly, peak levels around 1.5 to 2 hours, and cleared relatively fast too, half-life of maybe 5 to 7 hours. Importantly, no serious side effects linked to the drug, and no antibodies against it were detected.
Encouraging first step for an inhaled approach. What about studies in actual COVID patients?
Yes, there was another one by Kaitov and colleagues, published in 2023. They tested an siRNA called SIR7-EMKK46, different target. And this one was formulated with something called a peptide dendrimer to help delivery.
A peptide dendrimer.
Okay.
And this was a trial in patients.
Right. Randomized controlled trial. They compared low-dose and high-dose siRNA against standard care in people with COVID-19.
What were they hoping to see? The main goal?
The primary endpoint was whether the siRNA could stop people from progressing to moderate or severe disease. They looked at things like temperature, breathing, oxygen levels, CT scans.
And did it work? Did it meet that goal?
Unfortunately, it didn't significantly reduce the overall proportion of patients hitting those severe criteria. That primary endpoint wasn't met. But they did see some interesting things, like a reduction in viral load, the amount of virus, particularly in the low-dose group, and some positive signs on secondary outcomes. Things like headache and shortness of breath seem to improve more in the siRNA groups.
So not a home run on the main goal, but hints of benefit. Any downsides? Side effects?
They did report some adverse events. Interestingly, anxiety, including diagnosed anxiety disorder, seemed to pop up more often in the siRNA groups compared to the control group.
That's definitely something to note. Shows why careful safety monitoring is so vital.
Absolutely critical.
Okay, let's pivot to another huge area, cancer. How can siRNA fit into cancer treatment?
Well, cancer is often driven by specific genes going haywire, genes that promote uncontrolled cell growth or stop cells from dying when they should.
Oncogens, tumor suppressors.
Right. So siRNA offers a way to very specifically target and silence those faulty, overactive cancer-driving genes.
Makes sense. Any examples in clinical testing?
Yes. A good example is an siRNA called TKM-PLK1, or TKM-08030. It targets a protein called polo-like kinase 1, PLK1.
PLK1, what does that do?
It's really important for cell division. And it turns out PLK1 is often cranked up way too high in many different cancers. So shutting it down seems like a good strategy.
And have they tried this TKM-PLK1 in patients?
They have. There were phase 1 and 2 trials in patients with liver cancers, like hepatocellular carcinoma, that couldn't be operated on.
What did they find?
They reported some encouraging results. Some patients had their disease stabilized, meaning it stopped growing, and some even had partial responses where the tumors shrank.
That sounds promising.
There was actually one really striking case report from a phase 12 trial, a patient with a very rare advanced cancer called ACC that had spread in their abdomen. They got TKM-0803001.
Man.
And later, surgeons were able to remove the tumor. When they looked at the removed tissue, it showed near-complete necrosis. Basically, the siRNA therapy seemed to have killed off almost all the cancer cells, a really significant result for that patient.
Wow, that's quite dramatic. How do they get the siRNA, like TKM-PLK1, specifically to the tumor? Back to delivery again.
Exactly. Lipid nanoparticles, LNPs, are often used here too. The idea is that nanoparticles can sometimes accumulate more in tumors than in healthy tissues, partly due to leaky blood vessels around tumors.
The EPR effect, enhanced permeability and retention.
Precisely. Plus, researchers like Tomohiro have explored modifying these LNPs further, maybe adding cell-penetrating peptides to help the siRNA get inside the cancer cells even better once the LNP reaches the tumor area.
So smart delivery combined with targeted silencing. Okay, hit COVID, cancer. It feels like this siRNA approach could be really widely applicable.
It really could. The range of conditions being explored is quite broad. Think about hemophilia, for instance.
Hemophilia, how would siRNA help there?
There's an investigational drug called fitusiran. It's an siRNA given by subcutaneous injection. Instead of replacing a missing clotting factor, it targets something called antithrombin.
Antithrombin. That normally stops clotting, right?
Exactly. So by lowering antithrombin levels with fitusiran, the aim is to kind of rebalance the system and allow people with hemophilia A or B to form clots better, even if they have inhibitors to traditional treatments.
That's a clever workaround. Did it work in trials?
The ATLAS PPX trial looked promising.
Yeah.
It showed that when patients switched to fitusiran, their annual bleed rates dropped significantly. That could be life-changing.
Definitely. What about more common chronic conditions or other genetic diseases?
Well, there's inclisiran. That's an approved siRNA. It targets PCSK9 to lower LDL cholesterol, the bad cholesterol.
PCSK9 inhibitors exist as antibodies too, right?
They do, but inclisiran offers a different approach. And the ORION-3 study showed it works well long-term, keeping LDL down over 4 years. The really neat part, it's only injected twice a year.
Twice a year. That's amazing for adherence compared to daily pills.
Huge potential benefit for adherence, exactly. Though it's worth noting another trial, ORION-5, in patients with a very severe genetic high cholesterol condition called HoFH, didn't meet its primary endpoint for LDL reduction at day 150. So it might depend on the specific patient group.
Okay.
Yeah.
Still, very interesting. Other genetic conditions.
Yes. Lumasiran is approved for a rare disease called primary hyperoxaluria type 1, PH1. It causes kidney damage from oxalate buildup. Lumasiran is an RNA therapeutic that significantly reduces oxalate levels in the urine.
Helping protect the kidneys.
Exactly. And then there's hereditary transthyretin amyloidosis, HATTR. It's a devastating disease where misfolded protein builds up.
Causing nerve damage, heart problems.
Right. And there are 2 approved siRNA drugs for the nerve damage part, the polyneuropathy, patisiran and vutrisiran. And there's evidence, like from the Apollo B trial with patisiran, suggesting potential benefits for the heart complications too.
It's incredible. A whole class of drugs tackling these really tough genetic diseases. Any other areas bubbling up?
Well, people are still looking at infectious diseases. The REEF2 trial combined an siRNA, JNJ 7376399, with another drug for chronic hepatitis B.
How did that go?
It did lower the hepatitis B surface antigen levels during treatment, which is good, but it didn't achieve what's called a functional cure, basically clearing the virus long-term within the study period. So maybe part of a future strategy.
Still learning there.
And other areas too, like dry eye disease, there's tivanisiran in development. And potentially for NASH, that fatty liver disease, targeting genes like PNPLA3 or HSD17B13. The pipeline is quite active.
It really sounds like it. With all this potential, we have to talk about safety. What are the main concerns with siRNA therapies?
Like any drug class, there are things to watch. Common reactions with these types of oligonucleotide drugs can include things like injection site reactions, maybe headache, fever, sometimes hypersensitivity, standard stuff often.
Okay. What about issues specific to the way siRNA works?
That's where you get into on-target versus off-target effects. On-target means effects related to silencing the gene you intended to silence. Usually good, but if you silence it too much, or in the wrong tissue, could that cause problems?
I see. And off-target?
Off-target is when the siRNA accidentally silences other genes it wasn't supposed to, because their sequence is maybe similar enough.
That sounds like a big challenge to avoid.
It is. And that's why there's so much effort put into designing the siRNA sequences to be extremely specific, using computer models, lab tests, trying to predict and weed out potential off-target matches. Again, those targeted delivery systems help, trying to get the drug only where it's needed.
So careful design and precise delivery are the keys to managing safety.
They're absolutely crucial, yes. Continuous refinement is happening all the time.
This has been incredibly insightful. If we were to boil it down, what's the main message you want our listeners to take away about siRNA?
I think the key takeaway is that this isn't just theoretical anymore. siRNA therapeutics are a real, significant, and rapidly growing part of biomedicine. They offer this amazing precision to silence disease-causing genes at their source.
We've certainly seen that potential across infections, cancer, genetic conditions.
Exactly. And the progress in cracking those delivery problems has been huge. We're seeing more and more of these drugs moving through trials and getting approved. It's a very dynamic field.
The examples really illustrate the versatility, targeting everything from cholesterol to rare genetic disorders.
They do. And of course, there's still work to do. Research is ongoing to make them even safer, even more effective, maybe delivered even more conveniently. But the trajectory is clear.
So maybe a final thought for everyone listening, thinking about this level of precision, the ability to target specific genes. How might this technology really reshape how we approach complex diseases down the road? What new possibilities and maybe even ethical questions does that open up?
That's the big question, isn't it? Definitely something to ponder. And if any specific area we touched on caught your ear, do look up the studies we mentioned, like Kaitov's or Chang's work on COVID, or the trials for fitusiran or inclisiran. There's a lot more detail out there.
Absolutely. Well, thank you for joining us for this deep dive into the world of small interfering RNA on the Biomedical Podcast.